Can you hear me? Okay. There's still six students is not in our class, but we will not wait for them, so we will start our today's class. Today is our mm, last the finally class of our theory course. Um, so today we will talk about the chromosome aberration. Um, As a class, we talk about the chromosome, and we learn the structure and some how to name the region of the chromosome, and mm, uh, and two forms of a chromosome, named the chromatin and chromosome. If some aberration or some the chromosome have some abnormality. For example, the number or the structure, then it will cause the chromosome distortion. Then, as we all know, the chromosome has the genes, is the carrier of the genes. So, if the chromosome have abnormality, then maybe one gene or even two or three, even more genes will be affected by this aberration. Then it will cause some disorders for human beings. Today, by taking today's class, I hope you master the concept, the types and formation mechanism of chromosome aberrations, and learn how to describe the abnormal karyotypes. You just need to learn the simple description method, because the abnormal karyotypes have simple and detailed description methods. You just need grasp the simple description method. Second, you should understand the research method of chromosome aberrations. As I said in the beginning of the class, the chromosome aberration can be have two types. One is the number aberration, another is the structure aberration. The chromosome aberration refers to abnormal changes of chromosomes in somatic cells or germ cells. So these abnormal changes include the number or the structure of the chromosomes. First, we will see as we learn about the gene mutation. We know the gene mutation has the spontaneous mutation and induced mutation. And in the nature, a spontaneous mutation is very, very rare. So, according to our experimental goal or some other aims, we will use some drugs or some chemicals to induce mutation. Here, the chromosome aberration also can be divided into spontaneous aberration and induced aberration. And the spontaneous aberration rate is also very, very, very rare, but it exists in our daily life. Because to some, for example, maybe you have no ignorance of it, and due to the induced aberration, it can be induced by some chemical factors or physical factors, biological factors, and these factors are the same to the gene mutation. But the chromosome aberration can be also induced by mother's age. So, the chemical factors, the drugs, the pesticides, the industrial poisons, and food additives, these chemical reagents we alters one by one. First, the chemical factors, drugs. The drugs include the anti-tumor drugs, or drugs to protect pregnancy and prevent pregnancy reaction, or some anti-convulsant drug. So, as you can see, the anti-tumor drugs or anti-convulsant drug, we need these drugs to protect us from disease, from the tumor or some other disease. But these drugs can also cause our chromosome aberration. So this is why when a woman is pregnant, she has better to know drugs even when she's ill. So the mother should protect herself better than to protect her children or her baby. And even the anti-tumor drug and the anti-convulsant drug, this drug maybe can help us to elevate the accurate the pain from disease, but these drugs also will induce the chromosome aberration. Next is a pesticide. Many chemical-synthetic pesticides can cause chromosome aberrations in human cells, and some organ-phosphorus pesticides can also increase chromosome aberration rate, such as the triclosan pesticides. So that's why we should eat some green vegetables and fruits with no pesticide. Next is the industrial poison. This maybe around us, we will not meet this industrial poison, but for the workers that work on the industry will meet them every day. So maybe these workers have more possibility to touch them. So these workers should protect them carefully. Eat some special clothes, or wear the mask, or wear the cap, and so on. Another finally is the food additive. Actually, except for the fruits or the vegetables that we eat some fresh fruit or vegetables. Almost all the fruits that we buy from the market or from have the food additive. So, but many food additive are rejected to the to our country. Standard. So all this the amount of the food additive have a normal standard, but if you eat this fruit every day, then the food additive will accumulate in your body. Then maybe this additive will over the standard. Some chemical substance contained in food preservatives and pigments and other additives can also cause human chromosome aberration, such as the EF2 or the cyclohexyl saccharin, and so on. So, if you want to protect yourself better, you should eat some food that by yourself, done by yourself. The second one is the physical factors, the ion radiation and the electromagnetic radiation. For example, some gamma rays, X-rays, and the CT. The third one is the biofactors. It includes the biotoxins and the virus. For example, the aflatoxin and the pathogen, the virus, for example, the rubella virus, the HBV, and the MERS virus. Last one is mother is. And if a woman is over 35 is she is called old mother. Then if he want to have her if she wants to have her baby, then she must do many many prenatal examination. This picture shows the maternal age and the Down syndrome. The Down syndrome, also known the trisomy 21, because the patients have one more chromosome 21 in her body. So here we from here's one has ovary will has a ovary is ovary is aging. Then the cells the ovary cells and the mitosis in the mitosis in the egg cells from the aging ovary will have some abnormality. Maybe it will not separate two daughter cells properly. Maybe one cell has one more chromosome, then the other have will lack a chromosome. Then it will cause some aberration of the chromosome. First, we will say because the chromosome aberration has a number aberration and a structure aberration. First, we will see the chromosome number aberration, number abnormality and its mechanism. And the chromosome number aberration includes the euploid and the aneuploid. First, the euploid changes. It will cause the triploid or the tetraploid. In the triploid, the patient's somatic cells have 3 sets of chromosomes with 1 additional chromosome in each pair. And so it will have 69 chromosomes in this patient. And for the tetraploid, the patient's somatic cells had 4 sets of chromosomes with 1 additional chromosome in each pair. There's the lowercase n. So this patient have 92 chromosomes in his body. Because the normal human beings have 46 chromosomes. Here, we will have a review on the chromosome set. So what is a chromosome set? We learn about the chromosome set in our last class, before class, the chromosome class. The chromosome set means the complete set of chromosomes contained in a normal germ cell, the gamete. So, we human beings have 2 chromosome sets, and each chromosome set have 23 chromosomes. So we human beings have 2 chromosome sets, and one from our mother and the other from our father. Due to the mechanism of the euploid change, first, it will form the triploid. It due to the dispermy or the digyny. As the picture shows, maybe 2 sperms into 1 egg cell. Then the karyotype is XYY with 69 chromosomes. The other will be the first 3 is also 2 sperms, the dispermy, 2 sperms into 1 egg cell. But the sex chromosome of the sperm may be YY or YX or XX. So the sex chromosome type may be XYY or XXY or XXX. The other may be the digyny. It means 1 sperm into a egg cell, but this egg cell has some abnormal in its in the meiosis II. So this egg will not only have a replication of the chromosome, but it didn't separate into two daughter cells. Then in the egg cells, it has 2 chromosome sets. So it may be one sperm may be the Y of the X into egg cells to form the fertilized eggs. So it may be the XXY or XXX. All this will cause the chromosome in human beings is 69, also named the triploid. The second one is the tetraploid. It because of the endoreplication, also named the endomitosis. It means the DNA replicates twice, and the cell only divides once. So this will cause this cell have 2 chromosome sets. Then when it forms the oxides, when it forms the fertilizer egg, then it will have 4 chromosome sets in these cells. Next, we will see the change in aneuploidy. In this condition, it have a hyperdiploid. It means one or more chromosomes are missing from a somatic cell. If it misses one chromosome, it names the monosomy, and if misses a pair of homologous chromosomes, it names the nolosomy. We will have an example for the monosomy. So, if it misses one chromosome, then in human beings we'll have 45 chromosomes left. From the Turner syndrome, because in this condition, the patients miss or lose one X chromosome. So his karyotype is 45X. This condition also named the Turner syndrome. If one or more chromosomes are missing, named the hyperdiploid. And if one or more chromosomes are actual in a somatic cell, it names the hyperdiploid. In this condition, have the trisomy and the polysomy. The trisomy means one pair of chromosomes has an extra one. For example, the trisomy 21, the trisomy 18, and the trisomy 13. It also will have two or more of chromosomes have it named the polysomy. This condition are common in sex chromosome. For example, the XXY or XXXY, XXYY or XXXXY or XXXXXY. All this because of the primary sex organ development or breast development or some stable fertility. This picture I saw the trisomy. One pair of the chromosome has an extra one. So the total chromosomes in these patients are 47. That is, number of chromosomes in cell is 47. So here is the trisomy 21. Shows the one chromosome 21 extra. Then this is the trisomy 18 and the trisomy 13. Due to here, you should pay attention that the 21 chromosome is the smallest oral chromosome in human. So in this case, we will learn hear about the trisomy 21, the Down syndrome in our daily life, but we rare hear about the trisomy 18 and trisomy 13 because these two chromosomes are bigger, then it will have more genes on them. If one chromosome is more, will cause the gene products will affect the balance of gene products. So many of these babies maybe die from the early of his development, then may be done with the bone. As to the mechanism of aneuploid change, one is the non-disjunction. It includes the mitosis non-disjunction of early cleavage of a fertilized egg. So in this condition, the fertilized egg from one cell become 2, 2 to 4, 4 to 8. So if this disjunction occurs earlier of the fertilized egg cleavage, then there will be more abnormal cells. If this condition occurs in very, very late of the fertilized egg cleavage, then maybe little abnormal cells will appear. The other is the chromosome don't separate during meiosis. As we all know, the meiosis have meiosis 1 and meiosis 2. So if the chromosome do not separate on the meiosis 1, then finally it will cause n plus 1 and n minus 1 cells. So all these cells are abnormal. And if the chromosome do not separate on the meiosis 2, then it will have half percent, the 50% of the cell is normal, and 50% of the cell is abnormal with n plus 1 and n minus 1. So here you should pay attention. If in the meiosis 1, the chromosome do not separate, then it will cause the daughter cells are abnormal. But if it happens on the meiosis 2, then half of the cells will be normal. Another cause is the chromosome loss. The centromere is not attached to the spindle filament. Then the splitting action is delayed. So, as we learn the structure of the chromosome, we know the chromosome has a centromere, and the centromere has a telomere. Then the telomere can attach to the spindle filament. Then separate the chromosome into two daughter cells when mitosis. But if the centromere is not attached to the spindle filament, then the chromosome will not separate to two daughter cells. So it will cause the chromosome left in the cytoplasm. Then it will be degradation, and will finally this chromosome is lost. Next, we will see the chromosome structure, aberration, and its mechanism. We have learned the number aberration, the euploid and the aneuploid, the triploid, the tetraploid, and the hyperdiploid, and the hyperdiploid, and the hyperdiploid. And its mechanism is non-disjunction or chromosome loss, and as the euploid is the dispermy, digyny, and the endomitosis. So you should grasp this. Next, we will see the structure aberration and its related mechanism. The chromosome structure aberration is the breakage of chromosomes first, and then the rejoin of the broken fragments. If a broken piece renews in its original place, it is called the renewal. In this condition, the chromosome returns to normal without causing genetic effects. But if the segments of the chromosome move to meet other segments or are lost, then it will cause the chromosome structure aberration. This is also known as the chromosome rearrangement. It occurs if a chromosome are not renewed or rejoined in situ. In the chromosome number aberration, here we should describe the description of this abnormal is with its total chromosome number and the sexy chromosome types. For example, here is 30 is 48 XXXY. So here is how to describe the chromosome number aberration with the total number of the chromosome and the sex chromosome types. But how to describe a chromosome structure aberration? Indicate the so first we should indicate the total number of the chromosome and the composition of the sex chromosomes, and then all this may be because it's not include the number aberration, so it is said in human beings it will be 46 XX or XY. Then use a letter or a triple character number. For example, the T means the translocation, and the delay means the deletion, or inv the invasion, to indicate the type of rearranged chromosomes. After that, then the first bracket indicates the series number of aberration chromosomes, and the second bracket indicates the region and the band number of the big point to indicate the break point. Later, we will learn how to describe them in detail. And here is the symbols and terms commonly used in karyotype analysis. We learned about this in our before class, the chromosome class. For example, the P, the short arm, Q, long arm, ins, insert, centromere, DEL deletion, inv inversion, DOP duplication, and so on. How to describe the chromosome structure aberration? Then we will learn them later. The clinical common chromosome structure aberration include deletion, DL, duplication, DOP, translocation, T, and inversion, INV, and so on. So you should grasp the deletion, duplication, translocation, and inversion. The chromosome breakage and rejoin of broken fragments are the basic mechanism of various chromosome structure aberrations. First, we will see the deletion, DL. As to the deletion, you should think the structure of the chromosome. So it may be the deletion may be occur in the terminal of a chromosome, and maybe in the interspace of the chromosome. So the deletion can be divided, include the terminal deletion and the intermediate deletion. This picture shows the terminal deletion means A, B, C, D, E, F, G, H. The AB is deleted, so it left the C, D, E, F, G, H. And the other maybe lose the CD fragment between the chromosome, so it left the A, B, E, F, G, H. It means the intermediate or interspatial deletion. Here, you should know how to you should learn the description method of the deletion. First is the terminal deletion. In this example, it occurs on the chromosome 1, and the fragment of the from the chromosome 1 from the Q21 region is lost. So from the Q21 to Q terminal is lost. Then this condition can be described as 66XX or XY, DEL means the deletion, and the first bracket is the chromosome 1, so it is 1. Then the breakage point is Q21, so the second bracket is Q21 here. So you should remember this sentence. The total number of chromosome, composition of sex chromosome. Then a symbol that indicates the type of ranged chromosome. Then first bracket indicates the serial number of aberration chromosome, and the second bracket indicates the region and band number of the breakpoint. So here, in our PPT, you should know it is 46XX or XY. Then DEL 1. First bracket is 1, and the other is Q21 or Q21. Another is the intermediate deletion. And the fragment from chromosome 3, the fragment Q21 to Q31 is lost. So in this condition, it can be described as 46 comma, SX or XY, then DL, first bracket, the chromosome 3, then the Q21 and Q31. Hello, can you understand this description method? Hello. Can you understand this description method?
Yes, sure.
Okay. Then, okay. Then we will see another type of the chromosome structure aberration is the duplication. Here, the duplication means the addition of more than one copy to a segment of a chromosome, resulting in the addition of one or more copies of genes to these segments. This maybe occurs between two homologous chromosomes because two homologous chromosomes have the same gene. And the duplication can be include the direct duplication and the inverted duplication. It is because the direction of the copy gene may be the same with the original, or it may be opposite to the original. In this picture, we can A, B, C, D, E, F, G. If the DE fragment is copied, then it may be A, B, C, D, E, D, E, F, G. And it can be also A, B, C, D, E, E, D, F, G. Then here, in first condition, it named the direct duplication. And in the second condition, it named the inverted duplication. So here, from our example, it occurs on the chromosome 2. The P14 to P23 fragment is copied. Then here, but this fragment is inverted duplication. Maybe in here has the P14 to P23 fragment. Maybe from the centromere here. From the centromere, here is the P14 and here is the P23. But the P14 is here and the P23 is here. So this direction is opposite to the original fragment. Then it described as the invert duplication. Here we should use the black one to describe them. The 46 XX invert duplication to P14 and P23. Now then use the detailed description method. The new chromosome composition is from the P terminal to P23 and breakage and rejoin the P14, then to the P23 and breakage and rejoin the P23, then to the Q terminal. So the detailed description method is used to describe the new chromosome composition. But the simple description just need point out the breakage point. Oh, here. It is no comma symbol. The second one is the inversion, abbreviated as INV. Inversion means the structural aberration caused by the recombination of segments inserted 180 degrees after two breaks in a chromosome is called inversion. Here, from this picture, from our example, from the first A, from the A example, it occurs on the chromosome 1. Then the fragment of the P22 to P34. Then it has the breakage, and then the direction of the fragment is opposite to the original. It's from the P34 to P22. Then this can be described as 46XX or XY INV on the chromosome 1, then P22 and P34. This inversion happens between the in the P arm, the short arm, so it names the paracentric inversion. And another example is the P15 and the Q21 fragment have an inversion. This can be described as 46XX or XY INV2 chromosome, then the P15 and the Q21. And this condition here, the short arm and the long arm also have a breakage. Then the fragment invert to the chromosome opposite. So it named the paracentric inversion because it happened between a short and a long arm. So the inversion can be include the paracentric inversion and the pericentric inversion. Then today the last type of the chromosome structure aberration is the translocation, abbreviated as T. It means a cleavage fragment of one chromosome transferred to another chromosome is called the translocation. It also can name the translocation invert. It is associated with change in gene location. As we all know, between two homologous chromosomes during the meiosis 1, it will have in the tetra period. In the tetrad period, it has an exchange during two homologous chromosomes, but non-sister chromatids. but the translocation occurs between two non-homologous chromosomes. So, if the translocation occurs, then the genes will change because it happens between two non-homologous chromosomes. Here, the orange one and the blue one, maybe the A, B, C, D, E, F, P, Q, R, S, T, U. Then have the A, B, C, D, S, T, U, and the P, O, Q, R, E, F. It has the translocation, has the reciprocal translocation. So it related with two chromosomes. And all the two chromosomes has their break point. Here we see an example of the reciprocal translocation. Here, from the chromosome Q21, then the chromosome 5 Q31. Finally, the chromosome 1 has a fragment of from the chromosome 5, and the chromosome 5 have a fragment from the chromosome 2. So this can be described as 46XX or XY translocation and 2 and chromosome 5. So it between two chromosomes. Use a semicolon. Write two chromosome 2 semicolon 5. Then in the chromosome 2, the breakage point is the Q21. Then with the semicolon in on the chromosome, the breakage point is a Q. Oh, sorry, a mistake. Q31. The translocation can be described as this. And in another, not also has this is a reciprocal translocation. It also has a Robertson translocation. This translocation occurring on the acrocentric chromosome. The chromosome 13, 14, 15, 21, and 22, because this 5 chromosome has a satellite. Because of his centric it has a acrocentric chromosome. It is there, so they have a satellite. In this condition, maybe two long arms of between these two chromosome can be connected together. Then the satellite, because the satellite is a heterochromatic, then it will no active gene expression. Then this will disappear from the cytoplasm. So later the two long arms of the chromosome, and it contains almost all the gene products of these two chromosomes. And due to this human beings itself, it will lost no gene production, so it has normal phenotype. But if this person want to have his own baby, then this will cause maybe this chromosome, this rejoin chromosome will look as one chromosome. Then the baby of them will be abnormal. So here, you should just grasp the simple description here. It is because they lost one chromosome, so the total chromosome number is 45, XX or XY, P, and chromosome 13, and semicolon 14, then P11 and Q11. This condition, the Robertson translocation occurs between the 13, 14, 15, 21, and 22 chromosome. So today we learn the chromosome aberration. It includes the number aberration and structure aberration. And the number aberration includes the europloid and aneuploid. And in the europloid is a triploid, tetraploid, and the aneuploid is a hyperploid and the hyperploid and hyperploid, and its related mechanism is the dispermy, the digeny, and endomitosis, and the non-disjunction of the chromosome loss. And due to the structural aberration includes the deletion, duplication, inversion, and translocation. Actually, the structural aberration has other types. For example, the ring chromosome and dicentric chromosome, and so on. So after class, you can search other types of structural aberration by yourself. In our class, you should grasp the deletion, duplication, inversion, and translocation, and their related mechanism between the because of the breakage and the rejoin of the fragments. So today's class is over. Do you have any question? Okay, so our theory course is over. Then on Thursday afternoon, we will have 2 experiments class. Okay?
OK，老师。
Okay. So
Thank you,
bye
老师。
bye. Bye bye.
拜拜。
拜拜。